Okay guys, so I will share my screen and we can start today's session. And when I share my screen, one of you can unmute and let me know if my screen is visible and if I'm audible. Can one of you unmute and please let me know if my screen is visible and if I'm audible?
Yes, ma'am.
Okay. Thanks. So, good evening, guys. Today is the 12th and the last session, not last, second last session of this session of these basically events where we basically discuss regarding genetic engineering, some sample assignment questions from last year. And in these live sessions, what we do is that we take some questions and we try to understand the concept behind it so that it's easier for us to solve similar type of other questions. Okay, so we'll start today's session with the first question for today. Sorry. So the first question for today is, which of the following subunits of type 1 restriction enzymes provide specificity for the recognition site? HSDR, HSDM, HSDS, and MOD. I'll wait for a few minutes. One of you can try and give me the answer. Sorry.
Ma'am, option C, HSDS.
Okay. Anybody else? Okay. So the answer will be yes, HSDS. Now, what are restriction enzymes? We discussed this a lot of times, but again, restriction enzymes are nothing but molecular scissors. And what are molecular glues? Ligases are molecular glues. Okay, so so nucleases basically and ligases have contradictory functions. So these endonucleases are nothing but molecular scissors. They can scan the DNA code, find very specific set of nucleotides and make a specific cut, okay, on both the strands. So what is a palindrome? They always pick a palindrome. So sequence that these restriction endonucleases they cut, these are mostly palindromes, almost. So what is a palindrome? A palindrome is something that is read the same way forward as backward. For example, if you read Hannah, Hannah is Hannah from forward and Hannah from backward as well. Same like level, madam, etc. Okay. So what does palindrome have to do with molecular biology? So palindromes in DNA sequences are very common. So genetic palindromes are similar to verbal palindromes. So the palindromic sequences in 3' base pair sequence is identical on both the strands. 5' and 3' N refers to the chemical structure of the DNA. Okay. So now each of the double strands of the DNA molecule is complementary to the other. So thus adenine pairs with thymine and guanine with cytosine. So that is how the pairing works between the two double strands of the DNA. So adenine and thymine, they have a double hydrogen bond, whereas guanine and cytosine have 3 hydrogen bonds. Now, restriction enzymes recognize and make a cut within specific palindromic sequences known as restriction sites in the genetic codes. This is usually a 4 or 6 base pair sequence. Example, so HAE3 is a restriction enzyme that searches the DNA molecule until it finds this particular sequence of 4 nitrogen bases. Okay. GGCC. See, okay. So this restriction enzyme, what it will do is it will scan through the entire DNA, okay, and it will find the sequence of these 4 nitrogen bases. So once the recognition site is found, HAE3 will go to basically cut the DNA. Okay, so this will generate blunt ends. If you can see, there is no chance of a sticky end over here. So generate a blunt end. So these cuts produced by scientists called blunt ends. Okay, because there are no 5' or 3' overhangs. So the ends are blunt. So these are blunt ends. So the names for restriction enzymes, they come from the type of bacteria in which the enzyme is found. So that is how the nomenclature is done. The order in which the restriction enzyme was identified and isolated. So EcoR1, for example, R, it's basically eco from E. coli, R strain of E. coli, so EcoR, I as it was the first E. coli restriction enzyme to be discovered, so EcoR1. So what are blunt ends and sticky ends? So remember how HAE3 produced a blunt end. So EcoR1, for instance, makes a staggered cut and produces a sticky end. If you can see, it basically cuts between the G and A bases of the DNA on both the strands, and it creates a 5' overhang. Okay, that is what it gives rise to sticky ends. Okay, so this is a blunt end. This is a molecular size if you see. This is a sticky end. Some more examples of restriction sites and of restriction enzymes with their cut sites. There is HIM3, which cuts between A base pairs. Then there is BAMH1, which cuts between GG. Then there is ALU1, which cuts between A and G, okay, on both the ends. So sorry, this arrows are a little haywire. Give me a minute. Okay. Now, why are sticky ends useful? So, DNA fragments with complementary sticky ends can be combined to create new molecules, which allows the creation and manipulation of DNA sequences from different sources. Okay. So, restriction enzymes have enzymatic activity. Obviously, that's why they are enzymes. They have a biological role. Definitely, it's diverse. There are a lot of different types. Diversity is there. Recognition sequence makes it more specific. Digestion conditions might also be different for different enzymes. The optimum temperature, optimum pH, all of these needed for any enzyme to work optimally to the best of its capacity might be different for different enzymes. Typical reaction mechanism or double and how double digestion occurs, we digest twice. All of these are important. Now, how does the enzyme activity work? So basically the enzyme first scans the entire sequence and then ultimately it reaches the recognition sequence and then it leaves through the recognition sequence. So what is the biological role of these enzymes? So the restriction modification system, restriction enzymes are paired with methylases. So what are methylases? Methylases are enzymes that add methyl groups to specific nucleotides within the recognition sequence. So this methylation prevents recognition by the restriction enzyme. However, the restriction enzyme within a cell does not destroy its own DNA. So that is how it works out, and it can destroy foreign DNA, but it will not destroy its own DNA. So how do you think the cell manages to distinguish between its own DNA and foreign DNA, because they will have the same base pairs almost? Okay. So how do you think the cell distinguishes between its own DNA and it does not cut or degrade its own DNA, and it identifies foreign DNA and it goes ahead to cut and degrade that? That is because of this modification system or the methylation system. So this methylases, they add methyl groups to specific nucleotides within the register sequence. This happens in the host organism. So in the cell, uh, that portion will be methylated. So that's why the recognition enzyme will not go and cut it. However, something that is coming from outside a foreign DNA will not have the methylation done and hence will suffer the wrath of the restriction endonuclease. So if we talk about the diversity, there is EcoR1, Escherichia coli R strain by 1. Okay, it cuts between G and A in the sequence GAA TTC. Then we have bacillar amylocoefficients by match 1. It cuts between G and G in GGA TCC. Then we have Wind3, Haemophilus influenzae. It cuts between A and A, GCCT. Then we have PST1, Providentius 240. It cuts between CTGCA, basically A and G. Then PME1, Pseudomonas mendocina. It cuts between T and A. So what can be the features of these regular sequences? They are mostly palindromic. Their length is might be 4 or 6. Okay, that is mostly the length of the regular sequences what the enzymes recognize. Site of cleavage, there might be sticky ends, 3' or 5' overhangs and blunt ends. The compatibility of the enzymes, multiple recognition sequences, isosomers, and type 2 versus type 3 restriction endonuclease. Now, if we talk about the digestion conditions, what are the most common digestion conditions for these? So, if we talk about the enzyme XPA1, it needs a buffer Tris MgCl2 and NaCl-DTT buffer at 20 degrees Celsius and also certain amount of BSA. Then you incubate at 37 degrees Celsius and then you can put 1 unit digest in 1 digest 1 microgram DNA in 1 hour, and you can heat inactivate the enzyme. Basically, you can inactivate the enzyme by heating it at 65 degrees Celsius for 20 minutes. Okay, so how does a typical restriction endonuclease reaction work? We'll have the DNA, obviously, a particular amount of the DNA. We'll have water, we'll have the reaction buffer, then we'll have the restriction endonuclease in particular quantity, like which we call units per microliter, and we'll incubate at particular temperature, particular pH for a particular time period, and tenfold excess enzyme ensures complete digestion. Okay, so enzyme should never exceed 1/10 of the reaction. BSA is often recommended because BAC is often recommended. Sorry. Because it stabilizes the enzyme, so a little part of BAC is added to it for the same reason. If we talk about double digest, so if we talk about double digest options, we can mix the enzymes in the same compatible buffers and conduct sequential digest. Some enzymes display star activity in certain buffers, which causes them to digest the DNA at sites other than the standard recognition site. Enzymes are proteins. They are biological catalysts that help them drive biochemical reactions. So enzymes names end with an ASE or endonuclease, right? Helicase, all of these. So bacteria have evolved with a class of enzymes that destroy foreign DNA. Okay, like for example, virus DNA. If there's a virus infecting a bacteria like bacteriophage, so the bacteria will have a class of enzyme that will destroy the foreign DNA and it will protect the bacterial DNA. So bacteriophages cannot multiply if their DNA is destroyed by their host. So if we talk about restricted nucleases, they restrict viruses because the viral genome is a foreign particle and foreign DNA it is destroyed upon entry. Then endonuclease means inside nucleus cuts the inside part of the nucleic acid. Exonuclease might mean that they are terminal cutters. They also recognize a short and specific DNA sequence and cuts it from inside. And a specific DNA sequence is called recognition sequence. So in 1952-53, Luria and Newman discovered the phenomenon of recession modification. So they named it as host-induced or host-controlled variation. Now, if you talk about restriction, bacteriophages varied in their ability to grow on different strains of E. coli. What are bacteriophages? They are basically viruses for the bacteria that target the bacteria. Now, once growth was achieved on one host strain, the phage could continue to grow happily on this strain. However, the phages were not restricted by the ability to grow on other strains as well. Then the nomenclature was like Smith and Nathan's proposed enzyme naming scheme. Okay, the three-letter acronym for each enzyme derived from the source organism. First letter from genus, next letters from species, additional letter or number represent the strain or serotypes. This is the official standard now. For example, the enzyme HIN3, HIN2 was isolated from Haemophilus influenzae serotype D. So these are the target sequences of some of the most popular enzymes. Now, endonuclease will cut DNA from the inside. It's not a terminal cutter. It's highly heterogeneous. It evolved independently rather than diverging from a common ancestor over here. And it's broadly classified into 4 types. What is the RM system? This is the restriction modification system. Its activity cuts foreign DNA at the recognition site. It has a methyl transferase activity as well that protects host DNA from cleavage by the restriction enzymes. So methylate one of the bases in each strand. So restriction enzyme is and its cognate modification system constitute the RM system. Now bacteria protect the self DNA from restriction addition by methylation of its recognition site. So methylation is adding a methyl group to DNA. So enzymes are classified based on recognition sequence and methylation pattern. If you talk about type 1 RE or nucleases, they are mostly multi-subunit proteins. They function as single protein complex. They contain 2 R subunits, 2 M subunits, and 1 S or specificity subunit. So you can cleave DNA at random lengths from recognition site. Type 3 is basically large enzymes. The combination restriction modification. They cleave outside of the recognition sequences and they require 2 recognition sequences in opposite orientations within the same DNA molecule. Type 4 is cleave only modified method with methylated hydroxymethylated and glucosyl hydroxymethylated bases. So restriction sequence has not been changed, but the cleavage takes place maximum at 30 base pairs away from one side of the sites. The sequence similarity suggests many more such systems in other bacteria and archaea. Type 2 is the most useful for gene analysis and cloning. There are mostly more than 3,500 arrays in type 2. They recognize a 4 to 8 BP sequences, and that is how the type 2 enzyme function. So each restriction enzyme always cuts at the same reduction sequence, okay? That is given. You can produce the same gel binding pattern. Many restriction enzymes are palindromic. For example, this. It's palindromic, so there will be a cut between G and A. Now, most enzymes, restriction enzymes, make staggered cuts. Staggered cuts produce single-stranded sticky ends or overhangs from different sources, okay? And they can almost easily be ligated. Now, some restriction enzymes cut DNA at opposite bases, so they leave blunt-ended DNA fragments. These are called blunt end cutters. So, discovery of enzymes that cut and paste DNA make generative engineering possible. So it's an enzyme that cuts DNA and generates fragments. And ligase basically the molecular glue joins the different DNA fragments. DNA fragments from different species can be ligated to create a recombinant DNA. An example of a plasmid. This is the values of a typical restriction digest, and these are some of the images from the paper. So half of the sequences is complementary to the half of the next sequence, and in DNA or RNA by which something a loop is also formed and it's called palindromic sequence, if you can see over here. Now, but the first identified are isolated from two different genes of E. coli, K12 and B. So these enzymes make random cuts in dsDNA at least 1,000 base pairs away from their recognition sites. So the recognition site is asymmetrical and is composed of two specific proteins, one containing 3 to 4 nucleotides and another containing 4 to 5 nucleotides separated by a non-specific spacer of about 6 to 8 nucleotides. So these enzymes are not multifunctional and are capable of both digestion and modifications activities. Now coming to the question topic, type of restriction enzymes possess the three subunits called HSDR, HSDM and HSDS. HSDR is required for restriction enzyme. HSDM is necessary for adding methyl groups to the host DNA methyl transferase activity, and HSDS is important for specificity of the recognition DNA binding successfully site in addition. So these are the most common available and used restriction enzymes. The type 2 enzymes vary from one type to the next. So they form homodimers with recognition sites that are usually undivided and are palindromic and 4 to 8 nucleotides in length. So recognition and cleaves the phosphodiester bond of double-stranded DNA at activity, and they usually require only magnesium plus as a cofactor. They can generate both sticky and blunt ends after double-stranded DNA cleavage. Type 3 recognize two separate mode palindromic sequences that are inversely oriented. They cut DNA about 20 to 30 base pairs after the recognition site, and these enzymes contain more than one subunit and require adomate and ATP cofactors for their roles in DNA. So DNA methylation and digestion. So type 3 enzymes are heterooligomeric and multifunctional. Their proteins are made up of composed of some foods. The proteins composed of two subunits, rest and modification. So restriction and modification. So the MOD or modification is required for modification while the rest is required for cleavage. And this type 3 enzyme recognizes short 5 to 6 base pairs long asymmetrical DNA sequences and cleave 25-27 base pairs downstream. So it's not exactly the same place. So these enzymes methylate only in one strand of DNA at the N6 position of adenosine residues. So newly replicated DNA will have only one strand of methylated, which is sufficient to prevent to protect against precipitation dilatation. So coming back to the question that we started with, yes, the answer is HSDS. Which of the following subunits of type 1 restriction enzyme provides specificity for the reduced sites? It's not HSDR or HSDF, it's HSDS. That's my answer. So I'll wait here for a few minutes. If you have any question, you can ask me, or we can move on to the next question after the few minutes. Moving on to the next question. So the next question for today is the release of DNA from DNA protein complexes after lysing the nucleus is achieved by the digestion with option A proteinase K, B DNase, C RNase H and D lipases. Which one do you think is the correct answer? I'll wait for a few minutes and discuss.
Ma'am, option A, proteinase K.
Okay. Anybody else? Okay, so the answer is option A proteinase K. So we'll talk briefly about DNA extraction and all of that. DNA extraction is a fundamental technique in molecular biology and genetics obviously. So it involves the isolation of DNA, deoxyribonucleic acid, from cells or tissues for various downstream applications such as DNA sequencing, PCR, genetic testing and molecular research, all of that. So DNA extraction is obviously a crucial step in many of the scientific and diagnostic procedures, and several methods are available for this purpose, right? So what are the principles of DNA extraction? First will be cell disruption. Obviously you need to lyse or disrupt the cells to break open the cells or tissues containing the DNA. So this can be done using various methods such as mechanical disruption, like grinding or homogenization, chemical lysis using detergents, or enzymatic digestion. Okay. Then protein removal. So after cell disruption, the DNA is typically mixed with proteinase K or other proteolytic enzymes to degrade proteins and remove them from the mixture. So proteins can interfere with downstream DNA analysis. So that's why proteins are removed from the mixture so that any downstream applications with DNA is not interrupted by the presence of proteins. Then DNA purification. Okay. So the next step involves the separation of DNA from other cellular components like RNA, proteins, and cell debris, and this is typically achieved through a combination of centrifugation, precipitation, or filtration methods. Then we have DNA precipitation. So DNA is often isolated by adding a salt solution like sodium acetate and a cold alcohol to the DNA solution. So this causes the DNA to precipitate out of solution as long stringy strands. Then we have washing or resuspension. So the DNA precipitate is then washed to remove any remaining contaminants and salts, and it is then resuspended in a buffer solution suitable for the intended downstream applications. Now, what are the common methods for DNA extraction? The phenol chloroform extraction. This method involves the use of phenol and chloroform to separate DNA from cellular components and yield high quality DNA. Salting out or salting in like methods. This method uses high concentration of salt like ammonium acetate to precipitate DNA. It's a relatively simple and widely used technique. And we have silica based DNA extraction, that is spin columns. So in this method DNA will bind to the silica membranes in the presence of chaotropic salts, and after several washing steps the purified DNA will be eluted with low salt buffer. So spin columns are commonly used for plasmid DNA purification and small scale DNA extractions. Then we have chillax or resin extraction. So this method relies on chelating or agent resin, so which will bind divalent metal ions and facilitate the release of DNA from cells. So it is nothing but a quick and easy technique often used for PCR based applications. Okay. Then we have magnetic based bead extraction. Magnetic bead based extraction. So magnetic beads coated with specific DNA binding molecules are used to selectively capture and purify the DNA. So this method is automation friendly and suitable for high throughput applications. Then we have the organic solvent extraction or CTAP. acetyl trimethyl ammonium bromide, which is used to lyse cells and separate DNA from other components. So this method is mostly used for plant DNA extraction. This is how phenol extraction occurs. So you add DNA and proteinase K in cells, incubated, the cell lysis will be there, and then you can add phenolchloroform, then vortex and centrifuge, and then you can transfer and retain the aqueous phase DNA. Schematic of silica based DNA extraction or spin columns. This is the calyx resin extraction. You add water, then you add the raisins, you boil and centrifuge, and you transfer and extract the DNA, retain the DNA extract. This is how the magnetic bead based extraction works. This is a sample plus magnetic beads. You mix and incubate, and then you separate. You use a magnetic rack to pull the magnet down, and along with it, the DNA will also get pulled down. And then you use various different salt concentration buffers to dilute the purified DNA. That is the principle of the assay. So you add the crude sample containing the DNA, apply magnet to isolate the DNA bound to beads, and then you wash DNA bound to beads and elute DNA from the beads. Now bacterial DNA extraction. So DNA extraction from bacteria involves first lysing the bacterial cells, typically with a detergent or enzymatic solution to release the genomic DNA, because we don't want contamination from genomic DNA. Following cell lysis, proteins and cellular debris are removed through a series of purification steps, often involving phenolchloroform extraction or spin column based methods, and the resulting DNA can be further purified and concentrated, making it suitable for various applications like PCR, DNA sequencing, or genetic analysis of bacterial genomes, etc. So, this process is crucial in molecular biology and microbiology research, enabling the study of bacterial genetics and gene expression. So this is schematic or a way to write down the procedure. That is how this works. So coming back to the question that we started with, release of DNA from DNA protein complexes after lysing the nucleus is achieved by proteinase K. Okay. Let's stop here for a few minutes and then we'll go to the next question. Okay, so moving on to the next question for today. So the human chromosome were digested with the ecoAD1 restriction enzyme. Large fragments released could be ligated in the dash vector, pre-digested with the same enzyme. Which one do you think is the correct answer? B28A plus, B28A minus, PBR322 and YAC or East artificial chromosome. I would wait for a few minutes.
Ma'am, option D, yak.
Okay. Why is it so?
Because yak actually carries a heavy genome, like human chromosome is quite heavier in molecular weight. So yak is good vector for that.
Okay. Yeah. So the answer is indeed YAC, which is East artificial chromosome. So, if you start from the beginning, we have discussed this a number of times, but again, vectors are DNA molecules that act as vesicles for carrying a foreign DNA fragment when inserted into host cell. So, plasmids are also vectors, right? So, cloning vector is a DNA molecule in which foreign DNA can be inserted or integrated and which is further capable of replicating within host cells to produce multiple clones of recombinant DNA. Okay, so the capability of autonomous replication. So vector should contain an origin of replication or ORI to enable independent replication using the host cell's machinery. And a cloning vehicle needs to be reasonably small in size and manageable. Large molecules tend to degrade during purification and are difficult to manipulate. So one of the advantages of being a small size vector is that it's easier to manipulate and it does not get degraded that's fast during purification. This is the sequence of a plasmid schematic. So you can see this is the ORI, the tetracycline resistance, which is the basically the selectable marker, the different, the multiple cloning site, the different uh, enzymes regulation site, and we have the ampicillin resistance as well. So presence of unique restriction sites. So it should have to allow cleavage of specific sequence by specific restriction endonucleases. So unique sites should be present on the vector DNA molecule either individually or as a cluster. That is what we call the MCS or the multiple cloning sites or the polylinker. Okay. Now, since there are many different restriction enzymes, it could be convenient to have a wide range of restriction sites in the vector. Solved by inserting a polylinker into the vector. So, this is what we call the polylinker or the MCS. It's a stretch of artificially synthesized DNA, about 50 base pairs long, which contains cut sites for several restriction enzymes. So, this allows for a wide choice of restriction enzymes. Many plasmid vectors contain an artificial region of DNA that has many different restriction enzyme sites. So such polylinker site is designed so that all the restriction enzyme sites in the polylinker are unique, and the corresponding enzymes not only cut the can only cut the plasmid once, because if it cuts it once many times, then it will be very confusing where to attach the gene insert, like the gene of interest at. So it should be ensured that the corresponding enzymes only cut the plasmid once. It would be easier to purify and handle. Plasmids are very easy to purify and handle, and there's minimum amount of non-essential DNA. And there are different types of cloning vectors as well that are used for different types of cloning experiments, be it amplification or expression. So the vector is chosen according to the size and type of DNA to be cloned, and also the purpose of the experiment. So it also depends on the host cell type, whether it's prokaryote or eukaryote. So if we start talking about plasmid cloning vector, they are self-replicating episomal DNA, double-channeled circular, that are maintained in bacteria as independent extrachromosomal entities. So all bacterial genera will have natural plasmids. Although they are not typically essential for bacterial survival, plasmids often carry genes and advantages under particular conditions. So, plasmids can range in size from less than 1 kb to more than 500 kb. So, each plasmid has an origin of replication to replicate inside the host cell, and plasmids are represented by 10 to 100 copies per host. So, they can carry DNA fragments as large as 10 kb as well. Now, the simplest cloning vectors and the ones that are most widely used in gene cloning are those based on small bacterial plasmids. So, plasmid has the ability to clone reasonably large, about 100 bp to 10 kilobase pieces of foreign DNA as well. So, initially, naturally occurring plasmids were used as vectors. So, naturally occurring plasmids are PSC 101, PSF 2124, COL E1, if you can see over here. However, naturally occurring plasmids, which are unmodified or non-engineered, they often lack several important features that are required for a high-quality cloning vector. So, therefore, naturally occurring plasmid cloning vectors have to be genetically modified, and these modifications include size of the plasmids are reduced, non-essential and toxin-producing genes are deleted, selectable marker genes are inserted like antibiotic-resistant genes, and then addition of the polylinker or the multiple cloning site is also included so as to give us an option to manipulate the plasmid to cut at different portions of the plasmid. So if we talk about PBR 322, this was designed by Bolivar and Rodriguez in 1977. That's why BR. Okay, and it's almost 4 kb in size, and it indicates P for P means plasmid, BR means Bolivar and Rodriguez who developed the plasmid. 322 is just a serial number. Now, what are the useful properties? The small size is less than 10 kb, so it's easier to purify, handle, and manipulate. The presence of two antibiotic resistant genes. Okay, so I think one of you was asking me in that last class. So here we have two antibiotic resistant genes which enables the selection of transformed cells from non-transformed cells and recombinants from non-recombinants by insertional inactivation. So whatever we do with blue white staining, we can do without the need of going into the blue white staining concept, because now we have two antibiotics, so we can make use of both the antibiotics, exploit those to not only select transformed cells from non-transformed, because only transformed cells will grow in antibiotic containing media, but also recombinants from non-recombinants. Okay, a reasonably high copy number. So generally there are about 15 molecules present in the transformed E. coli cell, and this number can be increased up to 3000 by plasmid amplification in the presence of chloramphenicol. It can take up to 3 kb of additional DNA as well. So this PBR 320 is derived from three different naturally occurring plasmids. The AMP-R or the ampicillin resistance gene from plasmid R1, the TET-R or tetracycline resistance gene from plasmid R65, and the original replication from PMB1. So what is the construction process of this PBR 322? The R1, the tetracycline, the ampicillin is in R1. If you can see over here, it is given, and from here we take the tetracycline. Okay, and then ultimately both of these are taken together, rearranged, and the fragments are ligated. The origin of replication is also added. So this was developed by several manipulations. It was produced by removing a 1089 BP base pair segment from PBR 322, and the deletion left the AMP-R and the TET-R genes intact. Okay. So the higher copy makes this vector more suitable if the aim of the experiment is to study the function of the clone gene. And the PUC vector is derived from PBR 322. It developed in the University of California in 1980, and there are several vectors in PUC series: PUC8, PUC9, PUC18, PUC19. So high copy cloning vector for replication in E. coli. It's constructed using the ampicillin resistance gene from the PMB1 and original replication from PBR 322. It's a high copy cloning vector for replication in E. coli. So the PUC vectors generally contain an origin of replication lag Z prime and an AMPR gene. So the MCS is located within the lag Z prime gene. It enables the selection of recombinants. Now PUC is suitable for blue by screening technique. So the LAGZ gene contains a cluster of unique restriction sites, and ligation of new DNA into any one of these sites results in the insertional inactivation of the gene and hence loss of beta galactosidase activity. So this is the key to distinguishing a recombinant plasmid, one that contains an inserted piece of DNA, from a non-recombinant plasmid that has no new DNA. Okay. Now PUC8 is descended from PBR322 and it is 2695 base pairs in size. So it contains an origin of replication, lag Z prime and AMP R, and there is no restriction sites within the AMP R gene. All the cloning sites are clustered into the lag Z prime gene. Now, what are the advantages of PUC? It's a plasmid having a copy number of 500 to 700 even before amplification, so it's huge. Identification of recombinant sites can be achieved by a single step process of plating onto agar medium containing ampicillin and X-galactose. That's what we call the blue wire screening. And clustering of the restriction sites, which allows a DNA fragment with different sticky ends to be cloned. So it can carry up to 10 kilo base pairs for nDNA. But what are the disadvantages? It cannot accept large fragments. Sizes range from 0 to 10 kb and the standard method of transformation are inefficient. Now you can also use viruses as vectors, right? So viruses are, anyway, they get into cells and they basically take over the molecular machinery, metabolic machinery of the host cell by inserting their own genome. So why not make use of that? So viruses can also be used as vectors. They're highly efficient for the cloning of larger DNA fragments, and the screening of the recombinant DNA molecule is easier. So the widely used E. coli phage vectors are lambda phage and M13 phage. So bacteriophage lambda, which infects E. coli, has been widely used as a cloning vector. It is a well characterized virus with both lytic and lysogenic life cycles. So lambda DNA circularizes within the bacterial cell and the DNA inside the phage particle is linear. So the size of the lambda genome is 48.5 kb. So in the lambda phage particle, the genome is a linear DNA molecule with two cos sites on each end. After the phage injects its DNA into the bacterial host, the DNA circularizes. So the two cohesive ends base pair and are ligated together by bacterial enzymes, so forming a circle. Now at each end there are complementary 12 base pair long overhangs known as the cos or the cohesive sequences at ends. So once inside the E. coli host cell, these pair up and the cohesive ends are ligated together by host enzymes forming the circular version of the lambda genome. Now lambda phage particle can accommodate up to 52 kb of DNA. So if 15 kb DNA is removed from the genome, then up to 18 kb of new DNA can be cloned. So the middle region of the lambda genome is non-essential and may be replaced with approximately 18 kb of the foreign DNA. Now the two classes of lambda phage vector. So in this type of vector, a large segment of the non-essential region is deleted and the two arms ligated together contains at least one unique RE site into which new DNA can be inserted. So insertion vectors offer limited scope for cloning large pieces of DNA. Replacement vectors, in which a central stuffer fragment or the non-essential DNA is removed and replaced with the foreign DNA. So they can easily carry larger pieces of DNA than insertion vectors, up to even 20 kb of inserted DNA. Then we move to cosmid. So cosmid is a hybrid of lambda phage and bacterial plasmids and behave both as plasmids and phages. So they are small multi-copy plasmid that carries lambda cos sites, ORI, MCS or multiple cloning site, AMP resistance, and can carry around 45 kilobase pairs of DNA to be cloned. That's huge. So it can easily be packed into a fudge. It's like a fudge with the coarse size, and they're able to replicate in the bacterial cell like a plasmid. And origin of replication allows to replicate in E. coli. So during the construction of cosmids, most of the lambda structure is deleted. Cos sites only remains. This is a modified structure. Almost 45 kilobase pairs of foreign DNA can be cloned. So what are the advantages? High transformation efficiency. It can carry huge segments of DNA, 45 kb. Easy identification on the selective media. Application and maintenance are easy. So M13 is a filamentous fudge with 6.4 kb of circular single-stranded DNA. So what is the life cycle of this M13 fudge? So injection of an M13 DNA molecule into an E. coli cell occurs with the via the pilus. Okay. So once inside the pili or the pilus, whatever you call it. So once inside the cell, the single-stranded molecule acts as a template for synthesis of a complementary strand, resulting in normal double-stranded DNA. So this process is not, this molecule is not inserted into the bacterial genome, but instead replicates until over 100 copies are present in the cell. You can see the schematic on the right hand side. So during the synthesis of new viral particles, the double-stranded DNA replicates by rolling circle mechanism to produce linear single-stranded DNA. So the linear single-stranded DNA then circularizes and is packed into the protein coat. Now, what are the several features of M13 that make it an attractive vector? So, genome is less than 10 kb in size, well within the range desirable for a potential vector. The double-stranded replicative form of the M13 genome behaves very much like a plasmid and can be treated as such for experimental purposes. Genes cloned with an M13 based vector can be obtained in the form of single-stranded DNA. Single-stranded DNA is useful for several techniques such as DNA sequencing and uh, in vitro mutagenesis. So the 6.4 bp genome in a genome in M13 has 10 closely packed genes, each essential for the replication. So there is a 507 nucleotide intergenic sequence into which only limited amount of foreign DNA could be inserted, and thus only a limited scope for modifying M13 genome. So construction of the M13 cloning vector. So LAGZ prime gene is introduced into the intergenic sequence. So the GG8-TTC sequence near the start of the gene is converted to EcoR1 site by in vitro mutagenesis. So M13 vectors are useful for producing single-stranded versions of clone genes. Only 1.5 K DNA can be efficiently, but the disadvantage is only 1.5 kb of DNA can be cloned, and single-stranded DNA is useful for several techniques such as DNA sequencing and in vitro mutagenesis. Then we move to Phage-Mix. Phage-Mix is a hybrid vector combining a part of the M13 genome with plasmid DNA. So Phage-Mix can clone DNA fragments up to 3 kb. Okay. So this can be made by this PMBL. It can be made by combining regions of M13 Phage and PUC8 plasmid vector systems. M gene from PUC8, LAGZ prime gene with MCS from PUC8, 1.3 kb fragment from M13 single-stranded DNA synthesizing region. So this is a schematic. If you can see over here, what is the conversion of the PMBL8 into single-stranded DNA. Now the host E. coli cells are transformed with PMBL8 and should be subsequently infected with the helper M13 phage to provide the necessary replicative enzymes. So single-stranded DNA fragments up to 10 kb in length can be cloned within the system. What is a high capacity vector system? So vector system that carries very large inserts. It's helpful for the analysis and cloning of complex eukaryotic genomes. And these types of vectors are used for creating libraries, genome sequencing, etc. Okay. Now we come to artificial chromosomes. So for large eukaryotic genomes, researchers often begin with vectors that can accept chromosomal DNA inserts of very large size. So most plasmid and viral vectors can accommodate inserts only a few times, few thousands to perhaps tens of thousands of nucleotides in length. So if a plasmid or viral vector is a DNA insert that is too large, it will have difficulty with DNA replication and is likely to suffer deletions in the insert. So analysis of the genomes of higher organisms requires the cloning of a much larger fragment than for bacteria. So therefore, larger DNA fragments require special vectors. This is the artificial chromosomes that have been developed to carry huge lengths of eukaryotic DNA. So what are the key components of artificial chromosomes? Centromere, which is required for the chromosome to be distributed correctly to daughter cells during cell division. Telomere, structures at the ends of the chromosome, which are needed in order for the ends to be replicated correctly and which also prevent the chromosome from being nibbled away by exonucleases. Then they are the origins of replication, which are the positions along the chromosome at which DNA replication initiates. Now these individual components are isolated, joined together in vitro, creating the artificial chromosome. So the types of artificial chromosomes are YAC, BAC, PAC, and HSC HAC. So YAC is the East artificial chromosome developed by these people over here, and it's huge segments of DNA up to even 2 kb or perhaps 2 million base pairs can be 2000 kb or 2 million base pairs can be carried on by YACs. So it's essentially a P32 PBR32 like plasmid into which a number of East genes have been inserted, and three genes URA3, SUP4 and TRP1 function as the selectable markers. So CN4 is the centromere sequence of the chromosome. Telomeres is provided by the two sequences called TEL, and ORI is the bacterial origin of replication and ARS is the yeast origin of replication. Okay. Selectable marker is SUP4 into which new DNA is inserted for cloning. So this is schematic of the cloning procedure if you see over here. It's obviously not drawn to scale. It's a chromosomal DNA is much larger than a YAC vector. Now the YAC has two forms, a circular form for growing in bacteria and a linear form for growing in yeast. The circular form can be manipulated and grown like any other plasmid in bacteria since it is the original of replication of bacteria and an antibiotic resistance gene. In order to use this in yeast, the circular form is isolated and linearized such as that the yeast telomere sequences are on each end. So, what are the applications? Cloning vehicles that propagate in eukaryotic host cells as eukaryotic chromosomes can be routinely used to clone 600 kilobase pairs of fragments in special types can handle DNA up to even 1.1400 kb length, allows to study the functions and expression of genes that are very long. Under some circumstances, YACs can be propagated in mammalian cells, enabling functional analysis to be done in the organism into which the gene normally resides. And YACs are very important in the creation of gene libraries. Then we move on to BAC or bacterial arrested chromosome. They are developed from the F plasmid of E. coli. They contain inserts up to even 300 kilobase space or longer. So the ARIPE gene encodes a protein essential for replication of at ORIS. PRA, PRB, and PRC genes encode proteins required for the proper segregation of the vector into daughter cells. Now, chloramphenicol resistance gene and LAG-Z prime genes are the selectable markers. So, LAG-Z prime contains MCS into which the foreign DNA to be cloned is inserted. So BACs have low copy number origin of replication and allow replication of clones at one copy per cell. So the DNA molecules are introduced into the host cell by electroporation. Then you have the P1 derived additional chromosome or the PAC, which is another cloning vector used for large eukaryotic DNA segments. PSC combine the features of bacteriophage P1 vectors and BACs. So packs of insert capacity in range of 70 to 300 kb, usually 150 kb. Now packs have a low copy number origin of replication, allow replication of clones at one copy per cell. And rDNA molecules are introduced into the host cell by electroporation. So vector with foreign DNA to be cloned are ligated and even packaged into phage and allowed to infect E. coli. HSC is a microchromosome with 6 to 10 megabase pairs in size and able to carry large DNA segments. Now, human artificial chromosome is a microchromosome that can act as a new chromosome in human cells. Instead of 46 chromosomes, the cell could have 47, with the 47th being very small, roughly 6 to 10 megabases in each size, and able to carry new genes. So, they are useful in expression studies as gene transfer vectors and are a tool for elucidating human chromosome function. So HSC is considered as a promising system for gene delivery and expression with the potential to overcome many problems caused by the use of viral based gene transfer systems. Now there are two approaches for the construction of HSC, bottom up and top down. So bottom up approach is also known as the assembly approach. So it strategy involves the de novo construction of HSCs by introducing necessary DNA elements for the maintenance of chromosome function into cells. It involves generating HSCs by introducing defined chromosomal sequences including telomeres, centromere, and origin of replication, etc. Another top-down approach, also known as TACF, telomere-associated chromosome fragmentation. So top-down construction refers to the truncation of natural chromosomes into smaller sizes. It involves modifying natural chromosomes into smaller defined microchromosomes in cultured cells. So within the cell, HSC maintains only one copy per cell. In 2001, human adult chromosomes called 21 HSC was reported and is based on a stripped copy of human chromosome 21, producing a chromosome 5 MB in length. Okay. Then we also talk about shuttle vectors. It is possible to insert two origins of replication into plasmid, and one for E. coli and another for the chosen host, any other host organism. So such vectors is able to replicate in E. coli using one origin of replication and in chosen host using alternative replication origin. That's a huge advantage. Such a vector is called shuttle vector because it can be transferred back and forth between two different species, bacteria and yeast, or mammalian cells and bacteria. Shuttle vectors can replicate into different organisms and have the appropriate origins of replication. Hence, one can clone a gene in bacteria, maybe modify it or mutate it in bacteria, and test its function by introducing it into yeast or animal cells. So, the first such vector was derived from fusion between the E. coli vector BBR322 and the Saccharomyces plasmid PC194 and PUB110. So in order for a shuttle vector to grow in both yeast and E. coli, it must have several essential elements like two origins of replication as we talked about, this and this. One for E. coli and one for yeast, so that the centromere sequence so that it is partitioned into the daughter cells during yeast replication. Selectable markers for both yeast and E. coli and a multiple cloning site for inserting the gene of interest. Expression vectors. Once a gene has been cloned into a vector, it may or may not be expressed. If both structural gene and promoter were cloned on the same segment of DNA, the gene may well be expressed. On the other hand, if only the structural gene was cloned, then expression will depend on whether a promoter is provided by the plasmid. Now, the basic features of expression vectors, it should have an effective and regulatable promoter for transcription. It should be translational signal sequences like ribosome binding sites, start codon, etc. And high copy number. So, more the copy number of vector, more the amount of clone gene, and hence more the product formation. So, as expression vectors contain sequences upstream of the clone gene, control transcription and translation of the clone gene, the expression vector shown uses the lac promoter, which is very strong but inducible. To stimulate transcription, an artificial inducer molecule called IPDG is added. And IPDG binds to the lac-I repressor protein, which then detaches from the DNA. This allows RNA polymerase to bind. Now, before IPDG is added to the culture, the lac-I repressor prevents the clone gene from being expressed. So the cloning vectors basically are molecules which are able to replicate several times in itself when a foreign DNA is integrated. So we have talked about all of these. So talked about bacteriophages, viruses that can infect bacteria. They are not able to infect eukaryotic cell. So they have a high significant mechanism for delivering its genome into bacterial cell. So it can be used as a cloning vector to deliver large DNA segments. If you can see, this is a schematic of how it forms. Host identification, phage attachment and injection of its DNA into the bacteria, lytic cycle or lysogen cycle, degradation of bacterial DNA and transcription of phage DNA, or phage DNA integrates in new phages can be released with or without survival of the bacteria. Replication of phage DNA and production of phage elements, assembly of new phages, release of new phages with destruction of the host bacteria. Then they have bacterial artificial chromosome. Okay. Each we have talked about each artificial chromosomes or yaks as well. Very large DNA fragments can be cloned in these yaks. So they're used for cloning very large DNA fragments, and their advantage over BAC is expressing eukaryotic proteins that require post-translational modifications. So yaks tend to produce chimeric effects which make them less stable compared to BACs. So chimeric is an organism composed of two or more different populations of genetically distinct cells originated from different zygote. So if you compare these two, YAC vectors are DNA constructs that are used for cloning DNA in yeast. BAC vectors are DNA constructs that are used for cloning DNA in bacteria. Now the length of inserted gene is almost 100 to 3000 kb can be inserted in these vectors, whereas for BAC it's much less. It's 75 to 300 kilobase pairs. In YAC, it can show chimerism. BAC does not show chimerism. Yeast machinery has post-translational mechanisms that are useful in expression of the eukaryotic proteins. Bacterial machinery does not have post-translational modification, but BAC is more stable in its defense and YAC is not. Then we have MAC or the mammalian chromosomes, which are still under development. They are microchromosomes that can act as chromosome in a population of human cells, which are also HACs. They can range from 6 to 10 megabase pairs that carry new genes introduced by human researchers. So they can be used as vectors in transfer of new genes. Studying their expression and mammalian chromosomal function can be elucidated using the microsomes in mammalian system. So you produce fragments of DNA using enzymes that cut DNA to specific base sequences. Link these fragments to self-replicating forms of DNA vectors. You replicate the recombinant DNA molecule in the host organism. and retrieve the cloned copies for use of modification, produce and purify gene product. So enzymes, that is all. That was all about yak chromosomes. Move ahead. Yeah. And it can replicate any inserted DNA via transfer to yeast cells as well. So these are the essential elements of yak. So coming back to the question that we started with, the answer will be yak. The human chromosomes were digested with the carbon digestion enzyme. The large fragments released could be ligated in the yak vector pre-digested with the same enzyme. Yes. The answer is yak. Okay. I'll wait for a few minutes. Anybody has any question, they can tell me, or we can go to the next question for. Okay, so next question for today. Tetracycline resistant gene produces the ribosomal protection proteins, beta lactamase, neomycin phosphotransferase 2, protophenical acetyl transferase. Which one do you think is the correct answer?
Ma'am,
Ma'am,
option
option
A,
A,
ribosomal
ribosomal
protection.
protection proteins.
Okay. Anybody else? Any other answer? Okay. So the answer is correct. It's ribosomal protection proteins. So if you talk about resistance, resistance can be occurring via various mechanisms. Either it is enzymatic inhibition, the enzyme is not working, or alteration of any kind of bacterial membranes, outer membrane permeability, inner membrane permeability, or whatever drug or antibiotic you add, it's rapidly the drug is ejected out or effluxed. Okay, or reduced drug influx, bypass of the antibiotic inhibition, alteration of target sites. So for example, you are targeting the ribosome, but its target site is altered. Altered cell wall precursor targets, altered target enzymes. All of these are mechanisms of resistance. If we talk about molecular genetics of antibiotic resistance, genetic variability is essential for microbial evolution. So it may occur in a variety of ways. Microevolutionary changes by point mutations in nucleotide base pair. Microevolutionary changes, whole scale changes like inversions, duplications, deletions, transposition, or acquisition of foreign DNA like plasmid mediated or bacteriophages transposons. Now, what are the mechanisms of resistance? Okay, so there are various, as I said, various, either plasmid can be effluxed out, the antibiotic altering enzyme can be working, antibiotic degrading enzyme can be coming into work. So if we talk about enzymatic inhibition, so the enzymes inactivating antibiotics. So beta lactamase is split amide bond of the beta lactam ring. So there are many types characterized by amino acids and nucleotide sequencing. So class A eventually hydrolyzed penicillins. Class B metalloenzymes have a zinc binding thiol group required for beta lactamase activity. Many beta lactamases are plasmid mediated, all that are produced constitutively. There are six main groups, those that hydrolyze benzyl penicillin. So beta lactamases, those that hydrolyze oxacillin and related penicillins. Carbene-penicillinases, and those that break extended spectrum beta lactams like aztreonum. Those that can break down oxy-mino beta lactams. All of these. So amino production of enzymes modifying antibiotics, aminoglycosides, chloramphenicol coded by plasmids, or even chromosomal genes. Okay. So modifying enzymes can be reactions can be N-acetylation, O-nucleotide dilation, O-phosphorylation, all of these. So chloramphenicol acetyl transferase, for example, inactivates the drug by 3-O-acetylation plasmid mediated. Then erythromycin ester is seen in E. coli hydrolyzed lactone ring, deactivating it, limiting utility of oral erythromycin in reducing the aerobic gram-negative flora of the gut prior to the gastrointestinal tract surgery. So degrading enzymes will bind to the antibiotic and essentially degrade it or make the antibiotic ineffective. So blocking enzymes attach side chains to the antibiotics that inhibit its function, like beta lactamases. So what can be the alteration of bacterial membranes? Outer membrane permeability. So outer membrane of gram-negative acts as a barrier to the antibiotics, especially hydrophobic ones. Inner membrane permeability, rate of entry of aminoglycosides into the bacterial cells, is a function of them binding to a non-saturable anionic transporter where they can retain their positive charge and are pulled across the cytoplasmic membrane by the internal charge of the cell. So this is an energy dependent process. So the energy generation or proton motive force may be altered through mutation. So promotion of the antibiotic efflux, it's a major mechanism for tetracycline resistance in gram-negative plasmid as well. Okay, and what is the bacterial system intrinsic capacity to restrict the entry of small molecules, especially gram-negative outer membrane is protective, gram-positive to outer membrane, hence more antibiotic sensitive. Okay. Now restriction of influx is a physiological way to reduce toxicity to bacterial cell. The most well-studied reflex system in E. coli is the ACR-AB Tol-C system, which comprises of an inner membrane protein ACR-B and an outer membrane protein Tol-C linked by a periplasmic protein ACR-A. So when activated, linker protein is believed to fold on itself, bringing the ACR-B and Tol-C proteins in close contact. Thus providing an exit path from the inside to the outside of the cell. So antibiotics are pumped out through this channel. There can be modification of target size as well. So alteration of ribosomal entry target size, familiar, it's like failure to inhibit protein synthesis and target cell growth. Basically, so affected antibiotics can be aminoglycosides, tetracyclines, macrolides, etc. Glycopeptides like vancomycin bind D-alanine, which is present at the termini of the peptidoglycan precursors, and the large glycopeptide molecules prevent the incorporation of the precursor into the cell wall. Okay. There can be alteration of target enzymes like the alteration of PPPs in beta lactams. Quinolones, for example, DNA gyrase is made up of GYRA and GYRB genes. So mutations in one of the gene results in resistance. Then there can be bypass inhibition, development of auxotrophs. They have growth factor requirements different from those of wild strain. So these mutants require substrates that normally are synthesized by the target enzymes, and if these are present in the environment, the organisms grow despite inhibition by synthetic enzymes. Okay. So, what are the factors that promote drug resistance? Exposure to suboptimal levels of antimicrobial, exposure to microbes carrying resistance genes, inappropriate drug use, lack of quality control, inadequate surveillance, poverty or war, and all of these. So, this is a table showing the different targets of the different antibiotics, their mode of action, and what is the mechanism of resistance. So if we talk about tetracycline, the tetracycline resistance genes do produce ribosomal protection proteins that protect the ribosome from tetracycline. So there is a gene called TET-M which produces a protein that stabilizes the interaction between ribosome and RNA, tRNA, in the presence of tetracycline. Then there is a gene called TET-O which protects the ribosome from binding to tetracycline. It's a translational GTPase that's similar to elongation factor EFG. Then we have TET-W, which is a gene that can basically confer high level resistance to tetracycline. So these ribosomal protection proteins are a target protection mechanism that interact with ribosomes and are involved in resistance to many clinically important antibiotics. So the mechanism of action for all ribosomal protection proteins may be similar, as the amino acid sequence of these ribosomal protection proteins encoded by the other classes at least 40% similar to TET-M. So yes, coming back to the question that we started with, tetracycline resistance gene produces the ribosomal protection proteins. That is the correct answer. Anybody has any doubt? Okay, so if you don't have any doubt, we can conclude the session for today over here. Okay, so this is the last live session according to the schedule, but there will be one extra live session, I think, of 1 hour that will be told. So, if you don't have any questions, we can conclude today's session. Stop sharing. Yeah, thank you so much. Okay,
Thank you,
thank
ma'am.
you.